Super. Thank you, Beth. Can you see the slides okay? Are you okay? Okay. Well, great. Thanks for the opportunity to present on the Dragonfly Mercury Project at the North Coast and Cascades Network. I'm here today on behalf of my team from USGS, Park Service, and the Appalachian Mountain Club. And I'll be presenting findings from the DMP, a decades-long nationwide assessment that uses dragonfly larvae as indicators of mercury risk in national parks, where I'm going to hone in specifically on results from the 5 NCCN parks that have been active DMP participants on the San Juan Islands, Lewis and Clark, Olympic, Mount Rainier, and North Cascades. So first, to give a broad overview of why we do this work, right? So mercury is a contaminant of global consequence. Now, we've all heard of fish consumption advisories due to mercury, for marine fish like tuna or predatory fish and freshwaters like bass. Even Olympic has a fish consumption advisory for their mountain lakes for trout consumption. As a result of mercury emissions into the atmosphere, the inorganic form of mercury has the capability to distribute widely across the globe, right? And then deposit on landscapes and there be converted into methylmercury, which moves through food webs and is the most toxic form of mercury, inducing reproductive and neurological harm. So not only is it a global contaminant, it's highly toxic, and the global burden of disease, or the number of days living with the disease, a metric used in public health forums, is higher than both Parkinson's and hepatitis combined. Another important component is that mercury is the only element apart from radioactive elements with an international treaty. That's the Minamata Convention. It's legally binding. It's ratified by 128 some signatory countries in an effort to reduce mercury globally to avoid human health exposure. Now, the Dragonfly Mercury Project data are situated with a long-term data set that can feed into better tracking the effectiveness of these reductions. Further, in that same vein, the effects of mercury are especially concerning hazard in the face of climate change, given the risk of mercury exposure is intricately linked to ecosystem conditions and environmental drivers that influence its bioavailability. I'll speak to that in the next slide, but whether it's through precipitation events or wildfire or glacier melt, these global changes and ensuing drivers may profoundly impact the mercury cycle. The DMP is well positioned to track both those global climate changes and emission reduction changes due to the program's robust data set on mercury and dragonflies. So I alluded to these complicating pathways. They're complex, the mercury cycle, and there's a lot of management implications. So for those that aren't familiar, you have these emissions, right, that come from either coal-burning power plants or mines or waste burning incinerations that emit inorganic mercury into the atmosphere, largely as a gas. When it's deposited or in wet or dry deposition, so rain, snow, dust events, and then finds its way on the landscape to rivers, waterways, wetlands. There are certain microbial communities that exist in these particular chemical environments that then can convert this inorganic form of mercury into the organic form, which is the methylmercury, which is bioavailable, and then can move into and up food chains where it can express in high levels in top level organisms. So there are multiple places where these natural resource management activities can come into play, along with these policy decisions that aim to reduce the inorganic mercury emissions. But I just want to note that there's this disconnect between the methylmercury in the environment and this inorganic mercury that's emitted to the atmosphere. And that's largely because of this ecosystem component. So these landscape type processes, these biogeochemical drivers that exist in certain environments that convert that inorganic mercury into the organic mercury. And so that's a message that I'm going to thread throughout this presentation. So the DMP is a national scale project centered on Park Service lands. Over 140 some protected places have participated in the past decade or so. It's critical to understanding mercury contamination across the US. So we're not only gaining knowledge to, provide science to inform policy and resource management, whether it's about risk communication for safe water bodies for fishing or actual earth moving activities to implement restoration. But we're doing that in a way that engages communities, which a couple of the other presentations today have spoken to, where we include the public in hands-on, minds-on science to the collection of dragonfly larvae. These ideal mercury biosentinels are indicators of contamination because they are sedentary, easy to obtain, relatively speaking, compared to fish in the water, and embody a high trophic position. It's where the public comes in, and these citizen and community scientists, whether they're kids or local community groups, retirees, volunteers, student classrooms, et cetera, that participate to varying degrees in this process that are really key to the project's success, especially when we're talking about a nationwide scale of data collection. So specifically here in the North Coast and Cascades Network, we look at the participant type and across the 5 parks, and we see that really youth comprise, roughly 40% of the different participants that engage in this project through the data collection. So like the Youth Conservation Corps or Northwest Youth Corps or interns, whereas we also, see that about a third are participating by MPS staff only, and that may be in the case where there are less accessible sites or there are capacity constraints to engaging the public, or even in the case of COVID, where contact was more limited. So now I'll move into the assessment of the spatial data. So first we're going to look nationally, then network level, then park level, and then site level. I'm going to kind of speed through this. So I don't expect you to be able to read this y-axis here, but these are the 100-some protected places in a recent publication. But I am going to highlight specifically the NCCN units. These are park-wide averages, right? So Olympic here at the top resides kind of in the top 10th of these data, whereas the other 4 parks are in sort of the bottom half. Now we see extreme variability, right? So like about 135-fold difference between the highest average concentration to the lowest. So what does that all mean? Well, we can compare those dragonfly mercury values to fish and amphibian mercury values and see that there's a positive correlation, then use that relationship to develop this integrated impairment index, which basically compares these values to benchmarks for organism health. And we can see in this nationwide map here that the cooler hues are indicative of more lower risk, and then these warmer hues are indicative of higher risk. When we look specifically at the North Coast and Cascades Network, we see that about 60% of the site year data fall in that moderate impairment risk or higher, which suggests that top predatory fish may exceed fish consumption guidelines for human health. Now we'll look at the network and park level. So at the top right, we compare NCCN against the other 30 or so INM networks participating. We see that the average network concentration of mercury and dragonfly falls like in the lower, 40%, 2/5 or so of the data set. Then we drill in specifically to these 5 parks and denote that there's about a 2-fold range between the lowest at Lewis and Clark to the highest at Olympic. And so these are park-wide averages. This is an aggregation of all the sites that participated at that park. We can then compare to these impairment thresholds and also to the means and denote where we see some exceedances. But I just want to call attention to the variability that might exist from site to site and specifically see how, from 72 sites at Rainier to 1 site at San Juan Islands, where we are finding this where some sites exceed and some sites do not exceed. So this is probably the better way to look at the data, even though I don't label the sites here because that would be, there's over almost 100 sites. So that's super impressive. And it's a lot of work that's been conducted by the parks themselves. But I just want to call attention again to the variability, these different drivers that are influencing these mercury concentrations. At Rainier, these 72 sites, there's a 9-fold difference from highest to lowest. We can compare again with these thresholds and the means and see that some sites within a park exceed and some do not. And then I want to give attention to at Olympic, a 30-fold difference. This bar actually goes off into the severe impairment threshold, where we're looking at, again, a 30-fold difference. These sites are relatively close to one another. So again, this speaks to these different chemical environments that exist even in the same watershed. And notion rings true for these temporal patterns as well, where we have trends that increase, trends that decrease, trends that may remain unchanged. And these are sites where we have 5 or more years of data. So it's a question of source versus process, right? We know that across the US, deposition is decreasing of mercury, but we don't see this correlation between again, these exposures in the dragonflies necessarily. And instead, what we're probably looking at are these biogeochemical drivers that differ. But this is a relationship between deposition of mercury and dragonfly mercury. We don't see a relationship. So as I close out, I just want to call attention to sort of our menu of options, why parks might participate. They might place emphasis on public participation or are especially concerned about fish consumption advisories. We know in a management context that these different drivers like DOC, sulfate, phosphorus, nitrogen, pH play a role in that production of methylmercury, and this nationwide model that we're developing now will help inform, on a context-dependent basis. So these predictors vary depending upon where and in what type of environment you're in. So even though the drivers are similar, they enact differently. It may have a negative correlation or it may have a positive correlation. And also the data, DNP data, feeds into RSSs, so research strategies, other park planning efforts, where we look at conditions and trends, and we can denote good, fair, or poor based on the biota concentration of mercury across taxa within a park. And with that, just a huge thanks to the North Coast and Cascades Science Learning Network, who has supported this work over the past several years. This data would not be possible without you. And of course, the parks themselves are sort of DNP ambassadors and all the public that you've engaged, citizen scientists and community scientists in this effort. And I leave you with dates. As we move forward, we encourage continued engagement, new engagement from other partners, et cetera. And we urge you to reach out and attend our upcoming webinars and get on our DNP list if you're not already on it. Vote in your favorite DNP steward and et cetera. And I welcome questions if there are any at this time. Thank you.
Thank you so much, Colleen. That's really interesting, and to see the parks here in the context of the huge data set that you have. We have time for a couple questions if anyone wants to ask any in the chat or out loud. Still claps coming in. I'm curious about outreach for any like concerning levels. How has that been implemented in different ways through the parks participating in this project?
Yeah, often if we see elevated levels, the park will contact us, in many cases has contacted us, and then we look toward better defining what's driving those levels and what the concern might be for other taxa in the park, right? So that has amounted to different, say, new study proposals. So we're looking for funds and several different park units through existing opportunities to try to better get a handle on where the mercury is coming from, what we can do about it, how we can mitigate the risk. So it just depends. It's kind of case by case. Mm-hmm.
Yeah, just filling those gaps so you can understand what the real cause is. And there's a question in the chat from Allison. Would you see physical signs in amphibians or fish if there are elevated mercury levels?
Yeah, that's a good question. It might more amount to maybe not so much at the organismal or population level, right, at this scale, but maybe more cellular or physiological, right? So we might see like altered behavior or reduced reproductive success in fish or amphibians. So, at very high levels, it might be more evident, but at the levels where we're looking, we're looking at mesocosms and comparing against established benchmarks and noting changes, at a more tissue or cellular level. Hey,
Yeah,
I'm reading
yeah.
this.
Oh,
how does this work? Oh, sorry, go ahead. You can read it.
I'll read it out loud and you can answer it. Has this work
Sure.
led to more coordinated cooperation with state programs on letting public know of those fish advisories?
Yeah, so great question. We do have that established positive relationship between Dragonfly Mercury and Fish Mercury. It would behoove continued work to better co-locate fish and dragonflies in specific parks so that we're indeed matching the taxa. And then therefore, once that's established sort of on even like a park or regionally specific level, then we'd be positioned to better inform or work with, say, states with respect to fish consumption advisories, because we cannot currently say, because these dragonfly are elevated, most definitively your fish exceed these advisories. We're not there yet. Nationally, we can say the data suggests that these top predator fish might exceed, but it would require a more fine scale work at a park level or a region level. Yep.
That makes sense. I'll sneak in this last one from Scott. Are there commonalities between these sites that have high mercury levels that you've noticed, or even maybe if that's too broad across the nation within our area?
Yeah, great question. I don't know that I can even answer it. I can say for the high site in the NCCN, that's a unique site. We know definitively of a source there. We can say likewise, like we do see some geothermal activity in some of our other parks where we see high sites, likely pointing, to a more natural source, like at Yellowstone, for instance, right above the super volcano. But not so much. There's sort of not like that one catch-all answer that, oh yeah, like there's high pH there. Again, this kind of speaks to the different drivers that vary in their ability to predict mercury accumulation in different parts of the country. So that's where this model comes into play and speaks to that context dependency I spoke of, where some parameters matter more in different parts of the country and in different habitats. So it's not like an easy answer, unfortunately.
Well, that's great that you have such a huge, robust data set then. You could start to get at that.
Yeah, it's hard to fit it all in 10 minutes, but yeah.
Well, thank you so much for your talk. This is really interesting. I've been hearing about the Dragonfly Mercury Project for a while, and it's great to see it pulled together in 10 short minutes.
Thanks. It's a great opportunity. Appreciate it. Thank you.